Hi, hello viewers. Welcome back again to one more other lecture session from this channel. So in this lecture session, we will be studying regarding packaging of DNA helix from the molecular basis of inheritance in detail. So in our earlier lecture sessions, we have studied or we have learned that in majority of the living organisms, DNA acts as the genetic material. And here the specific DNA which acts as the genetic material, it has to pack into the interior side of the chromosome. So here prior to the packing of the specific DNA into the interior side of the chromosome, at first the specific DNA, it has to wrap around one specific protein complex. So here after the successful wrapping of the particular DNA around the protein complex, then after the specific DNA, it is capable to pack into the interior side of the chromosome. Is it clear? So let me write it on the board. In majority of the living organisms, DNA act as genetic material. So here the specific DNA which acts as genetic material, it has to pack into the interior side of the chromosome. So here prior to the packing of the specific DNA into the interior side of the chromosome, the specific DNA, it has to take the advantage of one specific protein complex. DNA which act as genetic material has to wrap around specific protein complex in order to pack into the interior side of the chromosome. Right? So here prior to the explanation of how the specific DNA it packs into the interior side of the chromosome, at first let us have a look on the length of the DNA. So here what length of the DNA in general it packs into the interior side of the chromosome. So here in order to evaluate the total length of the DNA in case of one particular human cell, here we need to take the advantage of certain parameters into the consideration. So here as we all know, all the human cells, they are diploid in condition. That means the chromosomal content is 2n. So here the haploid content of the human cell, it has around 3.3 into 10 to the power of 9 base pairs. So let me write it here. Haploid content of the human cell has 3.3 into 10 to the power of 9 base pairs. Is it clear? So here in order to obtain the diploid content of the human cell, here we need to multiply 3.3 into 10 to the power of 9 base pairs with 2. Diploid content of the human cell, it ranges around 3.3 into 10 to the power of 9 base pairs into 2, right? So once when we multiply 3.3 into 10 to the power of 9 base pairs with 2, here we obtain 6.6 into 10 to the power of 9 base pairs. So here we need to multiply 6.6 into 10 to the power of 9 base pairs with 0.34. So here what is 0.34 here? The distance between the two successive nitrogenous base pairs in case of the DNA double helix. Is it clear? So here the distance between the two successive nitrogenous base pairs in case of one particular DNA double helix, here it is nothing but 0.34 nanometers, or in other terms here we can consider it as 3.4 angstrom units. 0.34 into 10 to the power of minus 9 meters. So here we obtain 2.2 meters length of DNA. 2.2 meters length of DNA. And if you look at here, the size of the typical nucleus, the size of the typical nucleus, it ranges around 5 micrometers to 10 micrometers in diameter. Size of typical nucleus, it ranges around 5 micrometers to 10 micrometers in diameter. So here the length of the DNA which is present in case of the human cell, it is quite larger in comparison with the size of the typical nucleus. So here the size of the typical nucleus, it ranges around 5 micrometers to 10 micrometers in diameter, but whereas the length of the DNA which is present in case of one particular human cell, it ranges around 2.2 meters. That means the length of the DNA which is present in case of one particular human cell, it is quite larger in comparison with the size of the typical nucleus. Is it clear? And right now we will be studying regarding how the specific DNA it packs into the interior side of the chromosome in case of one particular prokaryotic cell in detail. So let me arrange the things here. And if we look at here the packaging of the DNA helix in case of the particular prokaryotic cells. So here, as we all know, in case of prokaryotes there is no availability of well-defined nucleus or complex nucleus. In case of prokaryotes, there is an availability of only primitive nucleus. So the reason why the specific DNA, it remains in the cytoplasm of the prokaryotic cell. And here the specific DNA which remains in the cytoplasm of the prokaryotic cell, it may remain in association with one specific protein complex which is composed of positively charged amino acids. Is it clear? So let me write it here. In prokaryotic cell, DNA which act as genetic material remains in the cytoplasm of the prokaryotic cell. And here the specific DNA which is having the negative charge, it wraps around one specific protein complex which is composed of positively charged amino acids. So here once when the specific DNA which is having the negative charge, once when it wraps around one specific protein complex which is composed of positively charged amino acids, and here it leads to the formation of what? One specialized structure in case of the prokaryotic cell which is nothing but nucleoid. Is it clear? DNA which is having negative charge wraps around the protein complex which is composed of positively charged amino acids. So here once when the specific DNA, when it wraps around the particular protein complex which is composed of positively charged amino acids, here it leads to the formation of a specialized structure in case of the prokaryotic cell which is nothing but what? Nucleoid. So let me write it here, nucleoid. And here nucleoid is a specialized structure which is observed only in case of the particular prokaryotic cell but not in case of the eukaryotic cell. So here this is regarding packaging of the DNA helix in case of the particular prokaryotic cell. So here though the packaging of the DNA helix, it remains very similar both in case of the prokaryotic cell and even in case of the eukaryotic cell. But here the packaging of the DNA helix in case of the particular eukaryotic cell, it remains much more complex in comparison with the packaging of the DNA helix in case of one particular prokaryotic cell. Is it clear? So let me arrange the things here. So as already mentioned, in case of eukaryotic cell, the packaging of the DNA helix in case of the eukaryotic cell, it remains much more complex in comparison with the packaging of the DNA helix in case of the prokaryotic cell. So here in case of the eukaryotic cell, the specific DNA which is having the negative charge, it wraps around one specific protein complex which is composed of positively charged amino acids like lysine and as well as arginine. Is it clear? So let me write it here. Specific DNA which is having negative charge wraps around protein complex which is composed of positively charged amino acids like lysine and arginine. And here the specific protein complex, here it is nothing but what? Histone octamer, which is composed of 8 identical subunits. So let me write it here. Histone octamer, which is composed of 8 identical subunits like H2A, H2B, H3, H4. And in addition, even it also contains one linker histone, which is nothing but H1 protein, right? So here the specific histone octamer, it is composed of 8 identical subunits. And here the specific histone octamer, basically it is composed of positively charged amino acids. But whereas the specific DNA which wraps around the histone octamer, it is having negative charge. Because of the presence of 2 dissimilar charges, it leads to the strong attraction. So let us assume all these are the 8 identical subunits. 8 identical subunits, right? And here it is nothing but what? Histone octamer, which is composed of 8 identical subunits. And basically, it is composed of positively charged amino acids. And on the surface of the histone octamer, in general, the specific DNA which is having the negative charge, it wraps out here. DNA, right? DNA, which is having the negative charge. So once when the specific DNA which is having the negative charge, once when it wraps around on the surface of the histone octamer, it leads to the formation of a specialized structure which is nothing but what? Nucleosome. Now here, you may get a small clarification. What length of the DNA sequence in general it wraps around the histone octamer? In general, the specific DNA which is having the negative charge, it wraps around the histone octamer. But here, what length of the DNA in general it wraps around on the surface of the histone octamer? So here the answer to this question is very simple. Around 200 base pairs of the DNA sequence in general, it wraps around the histone octamer in order to form a specialized structure known as nucleosome. In general, 200 base pairs of DNA sequence wraps around the histone octamer in order to form a specialized structure known as nucleosome. So here the specific histone octamer, it is composed of 8 identical subunits like H2A, H2B, H3 and H4. Then what is the role of the particular linker histone? So here the particular linker histone, it plays an important role for associating one nucleosome with the adjacent nucleosome. Is it clear? The particular linker histone, it plays an important role for associating one nucleosome in association with the adjacent nucleosome. So the reason why here it is termed as what? Linker histone. Is it clear? And in general, in case of one particular eukaryotic human cell, there is an availability of total 2.2 meters length of the DNA. Eukaryotic human cell. Eukaryotic human cell has 2.2 meters length of DNA. 2.2 meters length of DNA. So does the 2.2 meters length of DNA, does it packs in case of one particular chromosome in case of the eukaryotic cell here? So the answer to this question is no. As we all know, in case of the particular diploid human cell, there is an availability of 23 pair of chromosomes. 22 pair of chromosomes are autosomes, but whereas one pair of chromosome is allosomes or sex chromosomes. That means 23 pair of chromosomes, they are capable to yield 46 chromosomes. So let me write it here. "23 pair of chromosomes. 23 pair of chromosomes. Generally, they are responsible for yielding 46 chromosomes. 46 chromosomes. And if you look at here, the organization of the chromosome in case of one particular eukaryotic cell. So here, prior to the organization of chromosome in case of the eukaryotic cell, the specific DNA, it remains in naked form. Is it clear? The particular DNA, it remains in naked form. Then after, the particular DNA, it wraps around the histone octamer in order to form a specialized structure known as what? Nucleosome. Once when the particular DNA, once when it wraps around the histone octamer, it leads to the formation of a specialized structure known as what? Nucleosome. So here one nucleosome, it is capable to remain in association with the adjacent nucleosome by taking the advantage of a specific protein which is nothing but what? Linker histone protein. So here the linker histone protein, it is responsible for associating one nucleosome with the adjacent nucleosome. Is it clear? So in this case, once when many number of nucleosomes, once when they aggregate together, it leads to the formation of what? Chromatin fiber. Is it clear? So let me write it here. Many number of nucleosomes. Many number of nucleosomes. Once when they associate, they lead to the formation of chromatin fiber. Chromatin fiber. And here the specific chromatin fiber, once when it undergoes condensation, by taking the advantage of specific proteins like non-histone chromosomal proteins, here it leads to the formation of what? Chromosome. Is it clear? Once when many number of nucleosomes, once when they associate together, here it leads to the formation of what? Chromatin fiber. So here the specific chromatin fiber, once when it undergoes further condensation by taking the advantage of specific proteins like non-histone chromosomal protein, here it leads to the formation of a specific structure which is nothing but what? Chromosome. So let me write it here. Non-histone chromosomal proteins are responsible for the formation of condensed structure which is nothing but chromosome. And the condensed structure of the chromosome, once when it has successfully organized by taking the advantage of non-histone chromosomal proteins. So here the non-histone chromosomal proteins, they play an important role in regulating the condensed form of the chromosome. So here after the successful formation of the condensed form of the chromosome, so here the condensed form of the chromosome, it undergoes replication in order to yield the sister chromatid during the interphase stage. So here chromosomes are nothing but what? Chromosomes are the condensed form of the DNA which is most commonly observed during the mitotic stage of the cell division under electronic microscopic observations. Is it clear? Chromosomes are the condensed form of DNA, condensed form of DNA which is observed during metaphase stage of mitotic cell division under electron microscopic observations. Electron microscopic observations, right? Electron microscopic observations. So here in case of one particular typical nucleus, there are two specific regions and these may include heterochromatin and the other one is euchromatin. Is it clear? In case of one particular typical nucleus, there are two specific regions and these may include heterochromatin and the other one is euchromatin. So here what is heterochromatin and what is euchromatin? Heterochromatin, it is darkly stained and densely packed and it is transcriptionally inactive. And most commonly heterochromatin, it is observed during the mitotic stage, and whereas euchromatin, it is loosely packed and lightly stained and transcriptionally active. So here, this is regarding packaging of DNA helix from the molecular basis of inheritance in detail. And in the next upcoming lecture session, we will be studying regarding Michelson and Stahl experiment in detail. So here, I hope that this video will help you a lot. So if you like this video, just hit the like button and share it to your friends.